Hello, my name is Wesley Legant, and I'm a joint assistant professor in the departments of biomedical engineering and pharmacology at the University of North Carolina in Chapel Hill. And I'm really happy today to be able to tell you about some of our lab's work as part of the Beckman Young Investigator Program, as well as the broader role that the Arnold and Mabel Beckman Foundation has had in enabling our research and in allowing us to train the next generation of scientists. So in my lab, we build and design new microscopes to study living cells. And if we look at the origins of the field, it's hard to imagine a scientific instrument that has had a broader impact on science than the microscope. In fact, if we look back to the early 1600s, an instrument from Robert Hooke led to the description of cells and the founding of cell biology by describing microscopic pockets underneath a slice of cork. In my group, we build new instruments to increase the speed, the sensitivity, and the duration with which we can image living samples. And these new instruments can allow us to watch single molecules in action as they bind and undergo chemical reactions in cells, or allow us to peer into living cells and watch as proteins are trafficked between different subcellular compartments. However, despite these advances, the way in which we operate these microscopes has barely changed in over 300 years. A scientist still prepares a sample, places it under the lens, and then navigates to a region of the specimen where they like to take a picture and then pushes go. Now, this becomes a limitation if we consider the amount and complexity of data that modern instruments can generate, which can be three-dimensional, it can be multicolor, and it can evolve throughout time. So this complexity poses several challenges. First, how do you quantify and test different scientific hypotheses from this vast amount of information? And second, we often aren't interested in the behavior of all cells all the time, but frequently only in rare events that may occur within specific cells at certain times. So how do we study just these rare events in more detail? And this becomes even more important when you consider that long-term biological outcomes are often driven precisely by such rare or fleeting biological phenomena. So to tackle this challenge, we took cues from the rapid advancements in artificial intelligence and machine learning that are impacting so many different fields today. Specifically, we initiated a collaboration with another Beckman Young investigator, Andrea Giovannucci, to explore whether the same algorithms that are in use in self-driving cars might be adapted to create a self-driving microscope that could automatically capture images of specific biological processes. And as a first application, we trained an instrument to capture movies of dividing cells. And this is important because errors in cell division affect species ranging from fungi to humans. They're the primary cause of birth defects and infertility, and they're key drivers of cancer growth and progression. However, capturing high-resolution movies of this process under a microscope is challenging for several reasons. First, cell division is quite rare. Second, mitosis is highly dynamic and evolves in seconds. And finally, it's quite fragile. So the same light that we use to illuminate the cell to image it can be quite toxic and prevent the cell from dividing properly. So to accomplish this, we manually identified cells in different stages of cell division from our images, and then use these images to train an algorithm that can automatically locate and identify these rare cells automatically. So as the microscope scans throughout the specimen, the network automatically logs the position and mitotic stage of each cell that it sees, and at the end of this scan, we have information about all cells within our population. So for example, across several experiments, we can see that when imaging almost 500,000 cells, only 1% of them or less are undergoing cell division at any given time. And of these cells, cells in prophase, which is the earliest stage of mitosis, are the rarest, consisting of only about 1 out of every 5,000 cells. Now, this type of population-level analysis is useful, for example, in applications of drug screening, where we can see that even very low-dose treatment with Taxol, a common chemotherapeutic drug, reduces the percentage of cells in mitosis. Where real-time analysis becomes even more useful is that it allows us to program the microscope to react to what it observes within the images at rates that can be much faster than could be accomplished by a human observer. So for example, when the microscope detects one of these rare cells, we can program it to automatically switch to a different imaging modality and study these specimens in more detail. For example, by collecting three-dimensional multicolor images to better understand the organization of chromosomes, shown in blue, the microtubule spindle, shown in yellow, or the kinetochores in pink, which are protein assemblies where microtubules attach to the chromosomes to generate the forces required to pull the DNA into the two separate daughter cells. Alternatively, we can program the microscope to switch to a live imaging mode to capture dynamic movies of the cell division process. From these movies, we can then automatically identify cells that experience mitotic errors by identifying spatial outliers of kinetochores that deviate from the expected distributions within cells. So to summarize, as part of the Beckman Young Investigator Program, we've utilized machine learning to develop a self-driving microscope that can automatically analyze and respond to what it's imaging. We've used this instrument to automate high-content imaging of rare cells within a population across a large field of view, and to use live imaging to better understand the origins and frequency of cell division errors under both normal and drug-perturbed conditions. And now we're currently working on training the instrument to image other rare phenomena. For example, to image the process of an immune cell attacking a tumor cell. So I've shared with you the collective work of a team of researchers, many of whom will be starting their own research groups or transitioning to postdocs in related fields in the coming years. And one of the things that I've always been impressed with Dr. Beckman's career is that he was able to use a fundamental understanding of engineering principles to develop instruments that have had such a broad impact on a spectrum of diverse scientific fields. And I think that this really demonstrates a fearlessness to not remain stagnant, but to follow the most interesting questions and opportunities, regardless of where they may lie. One of the things I've been really grateful of the Beckman Young Investigator Award is it's given us the flexibility that even though we never know exactly what we're going to see when we look underneath the microscope, we have confidence that we can follow this into new research directions or initiate new collaborations that will be able to produce new findings for years to come. Thank you very much.